Okay. Perfect, perfect. Well, all right, everybody. I guess it's after 10 o'clock, so we should get going. Well, good morning. I will say that I'm going to be sorry to not have this trip into work anymore. So for the last 5 days, I've been getting up and taking a taxi in and enjoying my cup of coffee from the machine upstairs in the entryway, and I'm going to miss the routine. So I hope I get a chance to come back at some point in the future, and that, or at least buy a copy of the coffee machine to bring back home. So, well, okay. So here we are at the final day. I was going to today cover some issues that are related to the different kinds of neural coding that fMRI can measure, the direct effects, carryover effects, MVPA-style distributed representation, and think a little bit about what different information those measures can convey regarding neural representation, and then the perhaps surprising or counterintuitive ways that they can interact and produce results that are unexpected. Now, I know that there are some of you who are here today who were not here on Wednesday. And on Wednesday, we went through, at a slower pace, some of these ideas about different types of neural coding and carryover designs and how these things can measure it. So I'm not going to repeat everything from Wednesday, but I will, as we get going, revisit some of these ideas just to refresh everyone's memory about different kinds of neural coding. So that's what we're going to go through for this first hour, is the different types of interactions between adaptation and distributed pattern analysis, all the rest. We'll do that for about an hour and then take a break. And then in the second half, I thought it would be good we could just throw it open to some questions from the audience about different experimental design issues and fMRI topics. I'm happy to just kind of work with whatever you'd like to ask me about. If the room is silent, then as punishment, I will tell you all about de Bruijn cycles and ways of using graph theory to generate stimulus sequences. So start thinking of questions now, or else. Okay, so on Wednesday, we talked a little bit about carryover designs and as a type of experimental approach, and explicitly placed those designs within a heuristic, or within a set of ideas about different ways of measuring neural function. So one thing we talked about was that fMRI can be used, what I term the direct effects of a stimulus, right? Which is that if you could just measure, independently of anything else, what is just the pure isolated brain response to faces and the pure isolated brain response to houses, you might end up making a plot like that for a region of the brain called the fusiform face area. And so what you'd be saying is that the average bulk response of this piece of cortex of the fusiform face area is to respond more to faces than to other sorts of things. And of course, there are more sort of subtle kinds of relationships and functions that one might be interested in terms of the bulk amplitude of neural activity and some parameter of experimental interest. In the bottom here, I've just illustrated the contrast of a Gabor patch. But there could be other things, the speed of speech, the number of words to be remembered across a delay, and so on. So in any event, these are the average direct effects of a stimulus across a patch of cortex. We contrasted that kind of measure with the notion of a distributed pattern analysis, where you take the direct effect, that is, what is the isolated response to a given stimulus, but now consider that response at a per voxel level. So at each of these individual voxels, what was the response? And measure the direct effect of a stimulus across a set of voxels, and then look to see if the pattern is informative. And this is the fundamental basis of this entire world of multivoxel pattern analysis, classification techniques, machine learning-based approaches to interpreting fMRI data. Okay, so this is the pattern of direct effects. And that's sort of another thing that can be measured out. And then finally, in contrast to these first two measures, one can measure, using fMRI, the modulatory effect of one stimulus on the following stimulus. And this is the basis of within-voxel adaptation. The idea that if you repeat a stimulus that is similar along some experimentally relevant axis, that there will be a decrease in neural response to the repetition, and that decrease in response will be informative to some underlying theory about representation. So here's the simplest kind of design of such an experiment, where you have one stimulus you have a stimulus that repeats twice, and the integrated activity over time to those two stimuli is expressed in the bold fMRI signal, and it's lower for the when you have the repeated stimulus versus when you have a new stimulus. Okay, so in that we can describe as the modulatory effect of one stimulus on the next. And then finally, this is just the last couple of slides that we'd gone through on Wednesday before. We had talked about how these different measures, in theory, could be sensitive to different kinds of neural representation. And we talked about the possibility of fruit representation in the brain, right? So, if there was a patch of cortex that likes apples, you've got the face area and you've got the house area. So let's imagine we have the apple area, right? So a region of the brain that just responds more to apples than to anything else. Then you might have a set of voxels which contains neurons, all of which, or on average, has a net response to apples which is greater than to any other kind of fruit that you present. And so this would be measured using just that average direct effect. So that's a simple direct effect. You could imagine a different kind of model, which is where now you have different voxels are sensitive to different aspects of the fruit, such that the distributed pattern across voxels now codes for fruit identity. So in this case, the set of 4 voxels at the top happen to have different tuning for red things or yellow things, round or sort of oblong things. And so that pattern of activity across voxels is sufficient to distinguish the fruit. And so that would be like an across-voxel pattern that would be detected with the MVPA. And then finally, we considered that you could have a different kind of neural coding, which is where each voxel contains a population of neurons which are capable of representing the entire space of fruit. And so now it's just the pattern of neurons that are firing within a voxel that's responsible for coding the fruit identity. And to be able to distinguish these, you would need to be able to use within voxel adaptation. Okay, these are extreme cartoons, but you can imagine states in between, where there might be, for example, some population code that is distributed across the cortex within voxels, but then has, just enough of a bias, voxel by voxel, that it could support distributed pattern analysis and MVPA. And this is traditionally the argument that's been made for how MVPA can decode things like orientation selectivity in V1 cortex. This is the original Kamatani and Tong paper in Nature Neuroscience some years ago that started off the most recent MVPA enthusiasm. So that's the idea. You must have, for MVPA to work, some degree of distribution across voxels of differential sensitivity to some aspects of the stimulus. Got to be the case, otherwise there's just no way you could detect it with MVPA. Okay, so there's something about these slides which are very computationally intensive for the computer. It really struggles with the fruit. Okay, so we also talked about how continuous carryover designs, this one sort of a way of presenting stimuli in a counterbalanced order, can be used to measure all 3 types of this representation simultaneously. And I think even if you're not personally interested in doing carryover designs and using this approach, nonetheless, this is a didactically useful point that there are these different kinds of neural coding, and that techniques like this can allow you to examine what the relationship is between them. So even if you never wanted to do a carryover experiment, you might be interested to know that if you did MVPA, you might learn one kind of thing, and if you did neural adaptation, you'd learn a different kind of thing. And that's what we're going to explore a little bit. The core idea behind a carryover design is just illustrated here. It's a very simple equation. We spent some more time on Wednesday talking about it. But the basic idea is that the neural response in a voxel to a given stimulus is the sum of just the direct effect of the stimulus, what that stimulus would evoke in terms of neural response in isolation, as well as a term which describes the modulatory effect of any one stimulus upon any other stimulus. And that's really all we need to know for today to be able to move ahead, because the direct effect of the stimulus, that's what you can use for distributed pattern analysis, and this modulatory matrix, that can let you measure something that's of interest to those who study adaptation. Okay. Well, Well, so what I want to argue for the next, 40 minutes or so, is that there are cases in which you can get different sorts of results by looking at these different measures, that they are informative as to the type of neural coding that's being used for different stimulus spaces, and that also these two sorts of measures can be confounded or interact in ways that might not be initially obvious. So let's first talk about that interaction case. Okay, so to talk about this, we need to understand about multi-channel and norm-based encoding models. Okay, so yesterday, or on Wednesday, we had spoken a little bit about multi-channel type encoding models, but I'm going to reintroduce those so that we can then contrast that with norm-based. Okay, so these are two sorts of extreme models of how a population of neurons might represent things, the appearance of faces, types of fruit, different words, on and on. So here's the simple cartoon model. We might imagine that there's a patch of cortex. This, by the way, is the standard cartoon that I'm going to use for a patch of cortex that contains some neurons. So there might be a patch of cortex with neurons in a voxel, and that you have those neurons are responsible for representing a set of faces. And so these faces might be a little tough to tell, but they actually are different. They are of different appearance as you move across. In fact, what they are is a steady morph from one type of face to another type of face, so a smooth transition from one face to another. So one type of way that these neurons might represent the face is that there might be one neuron that is particularly tuned for this guy. Let's call him Bob. And so when Bob is on the screen, the neuron fires quite well. You know, there's another neuron that happens to like old man Steve over here, and so responds very well when he's on the screen, and so on and so forth. Other neurons which are ideally tuned to respond to other faces. And if you had a set of these neurons which have receptive fields with a bit of width that then tile the space of possible faces, well then you have the material for a multi-channel coding scheme. Indeed, this is the kind of coding scheme which has been examined with great success in representation of things like spatial position and generally in parietal areas, motion and direction of motion, MT. It's a simplified model, but one that has some pretty good evidence for a neural instantiation. So the basic idea is that when you present Bob, what's going to happen is the neuron that likes Bob the best will fire, but there will be other neurons close by which sort of like Bob and will fire sort of with a middle response. Okay? For this kind of neural tuning or this kind of neural coding, what I would argue is that adaptation is ideally suited to measure it. So the notion is that you measure the degree of reduction in response that's proportional to how similar two stimuli are as far as the neural population is concerned. In practice, what this amounts to is you're using adaptation to measure what is the distance between the two faces within a multidimensional space where the dimensions are defined by the number of neurons that make up the space, or perhaps the projection of that set of neurons on some simplified factorization. But without getting into those details, basically, there's some space, this idea of a multidimensional space in which any one face can be described as the simultaneous degree of firing of each of the set of neurons that make up that representation. Okay? And adaptation is giving you something about the distance between the two. And of course, there are many examples of experiments that have done this kind of thing. On Wednesday, I described some of my work which does this. I think one of the first or clearest examples of a test of this is a paper by Zhang et al. That appeared in 2006, where they had pairs of faces that differed in their similarity. So on one trial, they might present the first two faces, which are quite similar, but then on another trial, they'd present the two faces that are far apart. And what they observed was that there was a larger neural response to the different faces to the ones that were similar, and that supported this idea of a population code for similarity. Okay? Any questions about that as an idea? You can have a population code and you have a neural adaptation to measure it.
But that would be like the response in a single voxel. Well, in a area.
That's right. So in principle, you could measure this from a single voxel, right? But in practice, if the signal is small, you take the average over an area, and a typical practice is to say, well, the fusiform face area, I think that's kind of a homogeneous area of cortex, so I'll just take the average response. In about 15 to 20 minutes, if you can brace yourself, or hold on, we're going to talk about what heterogeneity across a set of voxels in adaptation might look like and the implications that might have. Okay, let's contrast this kind of representation with what we might call a norm-based code. So a norm-based code adopts a stance like this, and that is that there are a population of neurons that code not sort of in a multidimensional manner the similarity of one face to another, but instead explicitly represent how far away any one stimulus is from a stored prototype representation. So the idea is that we are all carrying around with us a representation of sort of the average face. Like we know what the average person looks like. And what these neurons are representing is how far apart or how far away from that average is the face that you're looking. So that amplitude of distance, in addition to there must be some vector representation as well, or angle, but mostly let's talk about that average angle distance, that the amplitude of firing, that's what's coding for how far the face away is from the average. So the idea is that as I've set it up, let's suppose that face down there, that youthful, everyman kind of looking guy, he's the average. But then as you get over here, this guy is a more extreme face, okay? So the notion is that you have this set of neurons that just have increasing firing for the more and more extreme the face is, okay? That's the whole model. So the basic idea there is that, of course, direct effects from fMRI can measure this norm-based coding in quite a straightforward manner. If you present a face that's from the center of the space that you're studying, then you get a small response. If you present an extreme face, you get a bigger response. Boom, that's it. And so really what is being proposed to be measured is the amplitude of firing is the length of this vector that connects the center of the stimulus space to wherever that face is in the space. That's the idea. So the appeal of these norm-based encoding models is that they seem to offer an explanation for some perceptual properties of faces and other sorts of stimuli. Most prominently, I think, they offer a nice explanation for caricature effects, right? So a caricature is where you take the appearance of someone's face and you enhance the difference that they have from average, right? So just classic, and there are a lot of very interesting effects. So for example, the caricature of a face is sometimes regarded as a better representation of the person than the person themselves. So like here's an example of this. This is a picture of Sarkozy, and you look at this, it's like, oh, it's Sarkozy. Like, of course, it's very easy to recognize. And that's actually, people are faster to recognize in general caricatures like this than the actual Sarkozy. So that's the sort of the caricature type manipulation. I must say he looks very fiendish here, doesn't he? He sort of. In any event, so that's the sort of the manipulation. And by the way, for U.S. audiences, I've got a great one of this of Bill Clinton. That's the other one I use. So so that's the caricature effect. And so there have been a number of studies that have tested the idea that there is this norm-based encoding representation for stimuli. So I think probably the canonical example is this paper by Loeffler et al. That appeared in Nature Neuroscience back in 2005. And so what they did was they took a series of these face-like stimuli, artificial face stimuli, and they had the faces which were either sort of very average or typical appearing faces, and then had progressive amounts of distortion or variation of the face, and then showed, using fMRI, that you could record an ever greater fMRI response from this fusiform face area for ever greater distance away from the center of the space. Okay? There have been other examples of this. There was a paper that appeared in 2010 by Panis et al. And so here now the center of the space, this is for a set of these two-dimensional popcorn-type shapes. The center of the space is in the middle here, and then these are the more extreme stimuli. And so what this plot purports to show is that from a ventral area that likes different shapes, that responds to shape, that there is a larger response to stimuli from the edges of the space than from the middle of the space. Okay, so this is norm-based coding. Well, okay, so this seems fine, right? I mean, these are two perfectly intellectually clear ideas about how neurons might represent information, right? You have a population code, you have a norm-based code. They're easy enough to define conceptually. These are coherent concepts. The problem is that multi-channel and norm-based mechanisms are confounded in measurement. When you actually go to try and measure these things and test one or test the other, you're actually measuring both. And so let me see if I can show you why that is. These are just absolutely confounded. And the reason is that there's an unavoidable context effect. Just to give you sort of the initial insight, the basic idea is what it means to be the stimulus at the center of a space means that everything else is more similar to you. Okay? If you're coming from the center of a stimulus space, you are more similar to all of your neighbors than to anything from the edge. And so then just to play that out. So consider, here's 5 faces that vary along a one-dimensional continuum. Okay? So that goes from face A to face B. And I will say, actually, that in all of the studies that I'm going to describe, and I'll put a little list up of them in a moment, this one-dimensional stimulus continuum is the standard, right? So you go from ba to da and morph in between, right? Or you go from face A to face B and get the morphs in between. These are the standard approach. So here's a set of faces that go from face A to face B. And now you have just a neuroscience experiment where you're going to present these 5 faces repeatedly, okay? And it turns out, actually, it doesn't matter for the point I'm about to make in what kind of ordering you present these faces. You could present them, one at a time. You could present them in blocks. You could use this carryover approach. It doesn't matter for the problem I'm about to describe. So let's suppose you want to measure adaptation, okay? And that is, you're going to measure, let's see, make sure I got this right. No, you want to measure norm-based coding. So you want to try and ask is there a bigger response to the stimulus at the extremes compared to the stimulus in the center? Okay, so you're going to go through your data, and after you've collected, and you'll take what's the average response to the center stimulus, and the average response to this stimulus, and the average response to this stimulus. Well, the problem here, and you might end up with a plot that looks like this, that there's a larger neural response to the extreme stimuli as compared to the center stimuli. Well, the basic problem, of course, is that the stimuli from the center were usually preceded by other stimuli which were more similar to it. Okay? So in general, the center stimulus would have been more adapted by other similar-looking stimuli, whereas, in contrast, the stimulus from the extreme edge tended to be followed by stimuli that were more dissimilar. So you can measure actually just the average transition size, the average, how on average, unlike the prior stimulus, each stimulus is, and that's what that plot looks like. So you would mistake an adaptation effect for a norm-based coding effect. And of course, the opposite is true. You can mistake norm-based coding for adaptation. So you might try and analyze the data and say, well, what was the response to a stimulus as a function of the stimulus that came just before? Okay? So you'd say, well, how much of a response was there to a 25% change in the stimulus, or a 50%, or a 75%, or 100? And you'd see a plot like this, and you'd say, good, I've found population coding. This is a recovery from adaptation. Well, the problem here, of course, is that it tends to be the case that for those transitions, which are the small transitions, those tend to include predominantly stimuli from the center of the space. I remember you had asked about this problem, I think, before, about how you might balance the sizes of transitions. In contrast, and I think this is the easiest to see, consider the biggest transitions in the space, the largest potential recovery from adaptation, where you make a big stimulus change, okay? Those necessarily involve the stimuli from the extremes, right? The only way you can get a 100% stimulus change is to use the stimuli that are on either edge. So as a consequence, these things are just completely confounded. You can't measure one and not measure the other. Yes?
But in multi-channel coding, you know exactly where the center of your stimuli are, because it depends on the stimuli you're presenting, which is the one that's more similar to all the others. Mm-hmm. But in norm-based coding, shouldn't you have like an absolute center that's independent of the stimuli you're presenting?
Yeah. So this is a great question. I think another of So the question is, hey, if you're using, in norm-based coding, isn't the model that you walk around with a lifetime's representation of the stimuli, and so you must have like a perfect absolute representation of where the center of the space is. And so now let's consider our stimuli with respect to that. That's right. I think this is another way to critique some of these norm-based coding studies is that isn't it surprising that for each experiment that does norm-based coding, the experimenters just happen to pick the perfect center of the space to correspond to that person's or that monkey's lifetime representation of the average. So I think that's your observation is an argument why norm-based coding models might be flawed. I think the rejoinder or the response is, oh, well, then what's happened is that the prototype, this norm, is relearned in the setting of the stimuli that are being presented. Okay? So maybe what's happening is you see some stimuli and you have now a flexible representation. Okay? And in a few slides I'm going to show you how the idea of a norm-based flexible center of representation is really the equivalent of saying you have neural adaptation effects that just go back a couple of stimuli. So I'll show you that intellectually those two things can be explained. They aren't, what we're building to is that these aren't really two separate things. These are one model with just a different time integration. That's what I'm going to argue. Yeah?
Do you also have a reference in the multi-channel? Because it's the center of the space. When you study adaptation, when you start computing the distances,
right,
there should be a 0-0.
No, there doesn't have to be. So because in these adaptations, the question is, well, hey, don't you also assume an absolute origin for the space in an adaptation type study? And you don't actually, because all you ever measure or care about is the relationship between the stimuli, right? So you could have this pair of faces, what's their relationship? Here, let's suppose this is the origin of face space, and you have these two faces that you're comparing. It actually doesn't matter if the two faces are here or here or here or here or here or here. All you care about is measuring is the relative distance between them. So actually the adaptation type studies don't require or generally make reference to an absolute center of, or an absolute origin for representation of the space. Do you buy that or not quite?
But you also have, in the norm base, you have a change of 25% between the center and the closest to the extreme, let's say. Mm-hmm. So there is also a change of 25%.
Yes.
From, let's say that we have 5.
Okay.
And the 3 is in the middle.
Right.
And the distance between the first and the second is the same as the second to the third, which
right.
is the reference.
Right.
So
I think we are agreeing. I think we are agreeing because my point generally is that in many experiments, the researcher takes a set of stimuli and says, I want to look at norm-based coding, and then takes the average response to each stimuli. And in other experiments, the scientist takes a set of stimuli and says, I want to look at adaptation, and looks at the relationship between them. And what I want to argue is that it is a mistake to think of these things in isolation.
yeah, exactly.
That's right. That you can't. It's not meaningful to think of each in isolation because the two sorts of the very hypothesis, the very idea of what you're trying to measure is innately confounded with the other thing you're not measuring. So I think we.
At the end of the day, it's a parametric modulation, both of them.
That's right. Well, yeah, that's right.
Which is the difference between the stimuli.
That's right. Well, yeah,
I mean, there could be some details. But
well, the reason I'm not saying yes, absolutely, is because the norm-based encoding studies explicitly ignore the size of the changes. They say, oh, that's, we're going to randomize the stimuli. That won't be a problem, right? We'll just randomize our stimuli, and then we're going to take a look at what is the response to each stimulus on average, okay? And I think we agree that that's a flaw.
yeah, exactly.
Yeah.
I just want to make sure I understand, because when you talk about them kind of being innately confounded, I
Yeah.
think you're just referring, though, to this kind of design where you assume, where you have continua, and not, and you have a center, right? And so not a design, for example, like a paired adaptation design, where you don't have a center, you're just looking at the difference between different pairs without assuming the center.
Well, let's see. So I guess the question is, could a paired adaptation design, suppose there was no actual adaptation, suppose adaptation didn't exist, okay? And you did a paired adaptation design, but there was norm-based coding, right? If you selected a group of stimuli that had, that varied between two morphs, right? That was a morph along a continuum, right? If you pick any two faces.
I guess that's the thing, you're assuming a continuum like that.
Yes.
Right, so that's where the problem lies, having that.
Well, actually, no. Let's suppose then you take a random sampling of stimuli from within face space. Okay? A random sampling of stimuli within face space will be distributed about a theoretical center point, right? So some of your stimulus pairs will be closer to the center and some will be farther apart. Oh, let's see, but would you end up then getting fooled by norm? I'd need to think through the math on. I'd need to think through that example. The problem is any time you use a single one-dimensional space, you're hosed, and all of the studies that I'm critiquing today have that as a property. It might be true that if you picked other sorts of stimulus spaces, you might be able to avoid this problem. In fact, and as I will argue, moving to multi-dimensional stimulus spaces tends to make this problem be less and less and less. So I think that's right. I think your intuition is correct. And we'll
A block design, you wouldn't have this problem either, right?
You would. Right. So, and there are all sorts of other problems. So, like a block. So, what sort of block design do you have in mind? Like you have.
You've got your center ones and you've got your non-centered ones.
Mm-hmm.
Right. And you see a bigger response for centered ones and your non-centered ones. I see.
Presuming, of course, that each block was presented in isolation in some way that one block wouldn't influence the next block, for example. Right. That's right. So yes, that's right. If you could imagine, if you could somehow get a measure where you bring somebody in on day one and show them just center faces, and then on day two you present them just edge faces, and then and then look at the differences between those, that'd be okay. But for reasons you can imagine, people don't do that experiment.
How long does the adaptation effect last for? And what if you put alternative stimuli in between the faces?
Right.
You're cleaning the...
That's right. It purged the system, right? So that's right. So one way of experimentally responding to this situation is you say, well, okay, I want to study norm-based encoding. So what I'm going to do is I'm going to do everything I can to block adaptation. And so you could do that. You could imagine spacing the stimuli out, putting in masking effects to try and block it. That's right, but you'd still have the experimental, like you'd need to convince yourself that you've managed to remove adaptation, and the only way one could do that would be by simultaneously measuring it. Yeah?
So, if putting in stimuli in between two stimuli or interests does not affect or does not remove your adaptation, then it would mean that if you have an experiment of 1 hour, that your activity at the end should be less than the beginning.
Oh, because you just did. That's that's interesting. Let's see.
And then we tried that.
What's that?
We have tried.
You have tried that.
Single cell, you see it's flat.
It's flat. Okay, so there is at least some degree of gain control in the system, so that there's not sort of a continuous decrement of activity over the course of an experiment. Yeah. No.
And if it's really if your stimulus is always affected by what comes before, then you would expect it.
If your stimulus was, let's see. No, not necessarily, right? What if it was the case that you can have? That's only if adaptation is purely a negative decrement phenomenon, right? So imagine that there's a mean response to a stimulus, and if you have two stimuli that sequentially follow, which are of average dissimilarity, then there's no modulation of the response. But if the next stimulus is more dissimilar, there's an increase, and if it's more similar, there's a decrease, then that would then produce a tonic level, right? So if it was the case that the modulatory effect was not actually all negative, but centered about 0, then that would not produce then a steady decline.
Then you say, let's say you have a face and then a house. That's putting in a house between two faces will remove your adaptation. Do I get that correct?
I guess I'm not claiming either way whether it would or it wouldn't, but I grant that's a model that we could discuss, right? So yes, okay, so maybe you put stimuli in between. I mean, there are long-term adaptation effects that persist over multiple stimuli and intervening things, the kind of things that Rick Henson and others have looked at. Yeah, so that's right. I mean, I think that it's quite plausible that there are these short-term adaptation effects which are easily disrupted by intervening stimuli or longer delays or things like that. Yeah, does that? For now, okay, all right. Well, so moving along. So this sort of, we've kind of fleshed out this didactic point in a recent paper, and really just the argument is that if you simulate some data, you simulate an experiment where you assume that there really is no norm-based coding, but you try and measure it anyway, or you assume there's no norm-based coding, but you try and measure it when there is adaptation, you get an effect that looks like adaptation, but it's just a spurious effect of norm-based coding and vice versa. So we show that just sort of thinking through this logical proposition in a simulation, you get that effect. And then we collected some data where we used, just again these sorts of shapes along a one-dimensional. We basically replicated the Panis experiment with, a one-dimensional array of stimuli, and found that you could find this ventral temporal region, which showed if you just analyze the data. So if you take the data that you collect and you analyze it just looking for adaptation effects or just looking for norm-based coding effects, you can find both. So if you analyze the data just looking for norm-based coding effects, you get smaller responses to the stimuli from the center of the space and larger responses to the stimuli at the edge. And if you analyze the data just looking for adaptation effects, you can find that. But then if you do this correction that I'm going to talk about, where you model both effects simultaneously, statistically, you can show that what looked like a norm-based coding effect in this region goes away. Okay? So this is what I'm going to get into, is that you can actually model these two things simultaneously and figure out which one is which. I will mention that this is a problem not just for studies of like norm-based and coding and things like this, but actually it's interesting if you're studying things about categorical boundaries. This is a bit of a concern too, because in general categorical boundaries are located at the center of a space, and so stimuli that cross categorical, that are close to a categorical boundary, are going to be different from stimuli that are farther away, and in any event. So, and I think there's some prior fMRI studies that have looked at sort of category boundary effects that might also have had this effect in it too. So, so here's just some examples. There's others as well of just studies in which a one-dimensional stimulus space has been used, right? And then the data have been analyzed to look for adaptation effects or for effects of norm-based coding, okay? So, of course, the key problem is, I would argue that a prototype is necessarily more average. And so, look, I'm not suggesting a radical reinterpretation of neuroscience. I think we just need a statistical approach to measure, like, hey, how much is an adaptation effect and how much is a norm-based encoding effect? This is a simple empirical question. You can just model the two and see which is which. And so what I've talked about, of course, is that these carryover designs are a way of doing this. I won't go through, I should have hidden some of these slides because we've already talked about it. But so just to kind of illustrate that you can do this, since I know this is a crowd that some folks do ERP work and MEG work, I thought you might be interested to know that you can examine this very set of issues well using ERP. So just for example, in a study that a student of mine, David Kahn, and I conducted, we used these very same stimuli. There were a set of 5. They came one after the other in one of these carryover designs. And the subject's task was to watch for a given target stimulus that appeared infrequently. And without getting into all the ERP details, because this is supposed to be an fMRI discussion, not ERP, we were able to identify sort of the different standard components that are described as being seen in response to faces. But then this is really the heart of it right here. So you can analyze the data or analyze the ERP data to code for each face what was the evoked response as a function of how dissimilar the face was from the prior face. And so then you can get just a set of waveforms, and you can see that there's a very nice parametric modulation of the degree of response as a function of similarity. So the greater the similarity, the more negative, the greater the dissimilarity, the more negative this P200 response was. And so, you can just pull out this one little piece and ask, okay, is there multi-channel encoding there? And the way we did that was, in the data, model both what was the response to a stimulus as a function of the prior stimulus. That's one set of covariates. And another set of covariates are, what was the response to the stimulus as a function of its absolute position in the stimulus space? Was it stimulus 1, 2, 3, 4, or 5? And so if you do that and then get out the beta values for the adaptation effect, boom, you still have an adaptation effect. Looks good, okay? So this would suggest then that, even after accounting for the fact that the larger transitions involve the stimuli from the extremes, you still get what looks like a monotonic change in this evoked response dependent upon sequential similarity, right? Conversely, you can take the very same data and analyze it looking for a direct effect. This is now like looking for this norm-based encoding. So the idea now is we're going to take what is the response, what is the waveform to each stimulus itself. So every time face 0 appeared, what was the response? Every time face 25, 50, so on. And you get this set of functions. Okay? And again, it looks like there's a bit of an ordering here, right? With the stimulus, which is the middle, 50%, up here, so that would be the center of the space, and the more extreme faces located down lower. So this is starting to look like a little bit of norm-based coding. So that's up a little bit closer. So if you take now just the heights of these peaks, you get something that looks like this, like, okay, looks pretty good. Here comes some norm-based coding, right? But let's do this now within a model that is also modeling for adaptation effects, right? Because maybe we're getting fooled by adaptation. So what happens to this effect once we model adaptation? Not so much. So at least in this one example, in this one case, we were able to show that there's an adaptation effect which is present and it cannot be accounted for by position within the space. And there was an apparent norm-based decoding effect that went away. So, and then ERP is the perfect environment in which to do this because you can get the response to every single trial not sort of smoothed out by the fMRI. So it's much easier to measure every response. Any questions about that claim? No? Okay. We're in the midst of doing similar sorts of things with fMRI. So you might remember yesterday, I started to show you some work which was along these lines with population coding of a set of faces. And we wanted to try and I showed you this just to motivate, like, oh, hey, we can measure this serial adaptation for similarity. This was on Wednesday. So you could contrast, again, this model of a population code. And I have some, oh, heck, these slides repeated. So, so once again, you can measure this kind of stuff. Oh, this is all old. These should have been skipped. But then you can also measure the contrasting possibility that you have norm-based encoding. Let me just skip a couple slides ahead here, because otherwise we're going to see a lot of repetition. So on Wednesday, what I showed you was that it was possible to measure this adaptation effect for a set of faces modulating over time. So we can do the same kind of things in these sorts of stimuli. We can look both at what is an adaptation effect and what is a norm-based encoding effect. So just to once again sort of set that up. Okay. So we have a set of stimuli that we know their mutual similarity, right? So here's a set of faces. We've done some behavioral testing so we know how similar one is to another. So one thing we can do is we can measure, hey, what's the response to a face as a function of how similar it was to the prior one? That's adaptation. But in this data, we can also simultaneously measure what's the effective distance from the center of this space, right? A putative sort of prototype or center-based coding. So then we just have some covariates. One is modeling an adaptation effect, and one is modeling a sort of norm-based encoding effect, okay? And so we have two data sets. We did this with two different sorts of sets of faces. And so what I'm going to show you here now is the effect of both the adaptation effect and this sort of prototype or norm-based coding effect, right? So this is the sum of those two. So you don't know yet, I haven't revealed yet, which one is how much is the adaptation effect and how much is the norm-based encoding effect. So I'm going to show you the sum of the two first. So we did this with one set of faces, and then we did the same experiment for another 15 people with another set of faces that look like this, that vary along different sorts of perceptual axes. So we just sort of generate the behavioral data and create this. So that was experiment 1. This is experiment 2. So we can get very similar kinds of results for these effects of adaptation and similarity, or of norm-based encoding. But then we want to ask, okay, what's the relative contribution of these two things? So, and we want to be able to examine this in both the experiments. So what we just did was we sort of combined the two, we took the two data sets, took the overlap between those two regions, and now pulled out these effects for each. So here's the interesting thing. In the ERP study, we found that there was a big effect of adaptation, small effect of prototype. In this data, we now find that for one set of faces, it looks like there's a big effect of prototype and a small effect of adaptation. That's kind of confusing. And for the other experiment, it's a little bit more of an even distribution between these two things. This starts to get quite confusing now, right? There are these two different mechanisms. It seems you use more one in one case and more one in the other case. Why would it be the case you'd have different kinds of coding schemes for stimuli that just vary subtly in their appearance? What I want to argue actually is that what's going on here is not two completely different coding schemes, but just a different period of integration over time. And so just a couple slides on this. For any of you who are going to the Society for Neuroscience meeting, I don't know if that's a meeting you guys tend to attend? No? Well, then you'll be missing a talk from a student of mine who will be describing this in more detail. Anyway, so with data like this, because of the way that they're ordered, we can actually test the idea that what a prototype is in this sense, what the center of the space is really just the sum of the recent history of the stimulus, okay? The idea is that for a given face, its response over like the last, if you sort of take how different was it from the face 1 back and 2 back and 3 back and 4 back, by the time you sort of integrate across a few stimuli going back, that now is the same as measuring what its distance is from the center of the space, okay? So we can do this in our data by modeling what was the effect of the last stimulus and 1 back and 2 back and 3, going back some distance. A technical detail is we model this with a Gaussian decay basis set, but that doesn't matter. So anyway, the whole point that I'm building up to is that what looks like a very different prototype effect, like a big prototype and a small adaptation effect, actually what's going on is that there is a decaying influence of the past history of the stimuli, okay? So that you have a big adaptation or modulation effect of the last stimulus, and then a bit of a smaller response to 2 stimuli ago, and a bit of a smaller response to 3 stimuli ago. And if you have just a little bit of a tail in history, if the neurons integrate over just a few stimuli back, that creates an effect that looks just like a prototype coding, because going a few stimuli back in the time now starts to converge on the center of the space. So to make a long story short, what unifies, I think, all of these different observations is that different stimulus spaces tend to lead the neural system to have different kind of temporal integration over time, to look farther back in time or less far back in time. And if the neurons tend to integrate information over a few more stimuli back, then you get what looks like a strong norm-based encoding state. But if they only encode back just 1 or 2 stimuli, you just see what looks more like adaptation. So that's the summary statement from all of this, without getting into much greater detail about that. Any thoughts, questions, or objections to that? Yeah.
So just to clarify, the n-1 is with respect to the same stimulus, the same phase, or to any phase?
To any phase. So on average, what was the modulatory effect of the stimulus, the last stimulus, right? And then what was the modulatory effect of the stimulus? One before that, and one before that, and one before that. So for each stimulus, the response, you're going to model the response to each face as a function of how dissimilar it is to the prior stimulus, that's one covariate, to 2 stimuli back, that's another covariate, and so on and so forth. So you look at what is the response to a stimulus as a function of its recent history.
But you also take into account that, or you also consider that in the similarity matrix, don't you?
The similarity matrix that I have described thus far is only for the immediately preceding stimulus. So it turns out a 2-dimensional matrix that describes all the interactions or all the effects of one stimulus, of a stimulus upon the next stimulus. A 3-dimensional matrix, that's 2 stimuli back. A 4-dimensional, that's 3 stimuli back, and so on. Okay? Yeah.
So do I get it correct that you bring everything back to a combination of adaptation and cross-adaptation and longer time?
I'm sorry, please ask that one more time.
Do you bring everything back to a combination of adaptation and cross-adaptation across a longer time?
Yeah, that's right. So I think in the end that what accounts for sort of in one experiment you see what looks like more of a norm-based decoding effect. In another experiment you see what looks more like an adaptation effect. I think what accounts for that is there's a single mechanism, which is you integrate over the recent history of stimuli that you've seen, right? So your response to a stimulus is modulated by how similar it is to recent things you've seen. And if that integration is for only very recent things, then that looks like what people describe as serial adaptation. But if that integration goes back a little bit further, then that starts to look more like norm-based coding. And the reason is that by the time you go back just a couple of stimuli, if you are sampling from all over the space, if you integrate over the last couple of things, then that's going to converge on the center of the space. So you can imagine an experiment which would directly test this, which is that you'd have an entire space of stimuli, and for a little while you'd sample from here, and then from a little while you'd sample from here, and then you'd sample from all over. And so what you should be able to show is that you could directly sort of show that the prototype is drifting, depending upon where in the space you're sampling. That would be the experiment, though, that would lend support to the argument I'm making. Okay? Yes?
So your claim is that how far back, how many n items back you integrate over depends on the context, so whether there's low or high variability.
Yeah, that's at least, well, I think that's our working hypothesis. I can't offer evidence for that right now, because to do that we would need to explicitly manipulate the gamut or range of the stimuli in a controlled manner. So we have a single space and then either make tight changes within a small context space or then large. That's our working idea, but I can't convince you of that right now. And I think there are reasons. This starts to get into the realm of like, well, what would adaptation be for? Or, so what are these neural mechanisms for? And I think that there's pretty good evidence in primary visual cortex of gain control and mechanisms like this to sort of normalize responses given the current variability and range of things that you're seeing. And so I think what we're seeing, what we're toying with the edges of are neural mechanisms that maintain sort of a homeostatic balance in a neural population, right? That kind of centers the range and variability of the firing and adapts it to the properties of the things you're looking at, right? Your sensory space. So crudely, so far, we're examining this, but I think that's the thing we're most interested in figuring out is how you might have adaptive mechanisms of firing that then tune neural response properties to the stimulus space that you're currently perceiving. So actually, last question then. How are we doing on time? What is it?
11, just before 11.
Just before 11. Okay, yeah.
So, do you consider the amplitude of the correlation between the two stimuli for the covariance that you...
This is not, let's see, so this is the amplitude of the correlation between...
You have the similarity matrix.
Right.
You tell me that you only consider the previous one, but you have a coefficient with the relationship between two stimuli.
Right.
Okay, so A and B.
Right.
So do you consider that coefficient for this? I mean, the amplitude of that coefficient for this relationship, for even if it's n2, n-2, n-3, n-4.
To be honest, I'm not sure I fully understand the question. So, okay, I got it. So we have a perceptual similarity matrix, which is only for a pairwise, right?
Yeah,
that's right.
But
Pairwise.
That's not
okay, so
temporal. I mean,
okay,
it's just the correlation between the stimuli.
Mm-hmm.
Okay. Do you consider that value to model this covariance?
Do I consider that value to model this covariance? Oh. Yes. Okay, good. I see what you're asking me. So, yes, that's right. We know something about how perceptually similar any pairwise combination of stimuli are, and we consider that an atemporal measurement. That's not something that's related to their order or timing. We're willing to assume that we know that as a property of the stimuli, and then we use that to look at what's the relationship of neural firing to then the stimulus history. So that's right, that one similarity matrix is used. Now, a much more sophisticated way of doing this would be to measure what's a behavioral modulation of response that trails back by stimulus history. And you guys remember that I talked originally about, on Wednesday, about, this approach comes from looking at tobacco leaves and blah, blah, blah. And there's this there's this literature which looks at sequential ordering effects upon magnitude estimation. That's the name of the literature. And that also shows that the influence of, so the results in that how far back, and by the way, this plot is not the ideal plot because we've got better ways of representing this now. The long and the short of it is, it looks like you integrate back over about 4 or 5 stimuli, and that varies a little bit between the two spaces. And that accords pretty well with what is seen behaviorally for sort of the recent history of stimulus integration in all sorts of domains, magnitude estimation, line orientation, all the rest, that there is a big effect of the last stimulus but smaller modulatory effects going back. Okay, that's probably enough to have baked everybody's head enough for one day. So let's take a break, and then when we come back, we'll do a question and answer, and I'll show a couple of slides about focal and distributed representation. And that is this idea that, hey, maybe multivoxel pattern analysis and adaptation could be sensitive to different things. So you've seen a few times now stimuli that look like these. These are, by the way, what are called radial frequency contours. That's the name of this type of set of shapes. It turns out that you can describe any closed 2D contour is sort of like a Fourier decomposition around a circle. So there's this mathematical way of making shapes, and these are radial frequency contours that have this property. And they've been studied by a number of folks in psychology. Cortese and Dyer are the ones who I think first started off with this. In any event, okay, so you have a set of shapes, and just like in many other experiments we've discussed so far, you can rate their similarity and come up with their pairwise similarity. And this was actually done then by Cortese and Dyer back in 1996, where they obtained the multidimensional scaling similarity for a set of these shapes and then sort of created a space. Okay, and so from that, again, as we've seen many times, one can create a stimulus similarity matrix. So I'm going to talk about an experiment that uses stimuli like this for which we know their mutual similarity. Okay, so here's the idea. So you've got this perceptual similarity matrix, but then, in a number of experiments that we've talked about, one might be interested in the neural similarity that underlies that. And so one thing that might be interesting is that the neural similarity might differ by region, and there's a number of sorts of experiments that have shown this kind of thing, and that's informative as to sort of the coding of the different stimulus space by that cortical area. But it's also interesting that it could be that you could have a difference of representation at spatial scale within a region. And so just to make that concrete again, so if you this is sort of the cartoon we've seen many times. If you have a set of voxels that have this kind of tuning, they're coarsely tuned to given sort of shape properties. Some neurons like oblong things, some neurons like tight turns, some neurons like broad turns. Then, a shape like that will produce one pattern, and another shape will produce a different pattern. As we know, that you could use a cross-voxel pattern analysis for that. But conversely, there might be neurons which are tuned to represent the entire shape space, in which case you need adaptation. So that's the setup. So it turns out that back in 2001, Hans Optebeek did a nice study, and I think he did this with Ruffin, didn't he? I think that's right. In which Hans and his colleagues made recordings from IT neurons to these different stimuli from the space. And he recorded from a set of neurons. So for example, from one neuron, he would present a set of these stimuli and make a recording of sort of what was the firing across that set of stimuli for that neuron, and then do the same thing for a second neuron, and a third neuron, and a fourth neuron, and so on, and then used sort of the similarity in the amount of firing across the set of neurons to essentially construct a multi-dimensional scaling solution of the similarity of the stimuli. And so the result was something like this, that he was able to find that the similarity of the stimuli as described by the population of neurons replicated the similarity of the stimuli as they were constructed and laid out. Okay? So that's sort of, again, a motivation that there might be something like this population code that we've talked about. So we've studied just these shapes in, actually not exactly Hans's shapes, but similar kinds of shapes in a paper from 2009 in cerebral cortex. It's the standard sort of carryover design. In this case, the subjects watched the shapes come by and judged if they were purple or red, and just had to press a button for each one. So there's two areas of the cortex that are of interest in this study: the ventral and the lateral occipital complex. So these are two posterior visual areas that have been identified for many years as being responsive to object properties. So the lateral LOC is located here on the lateral surface of the cortex. It has a very coarse but present retinotopic organization. And it goes by several names, LO1, LO2. There's other different ways of carving up this region, but generally gets called the lateral LOC. Now viewing the underside of the brain, this is the ventral LOC. It also has several names, VO1. It also gets called the posterior fusiform sulcus. So another region. And this area has even weaker retinotopic organization than the lateral occipital complex does. And both of these regions have been seen for a while as regions that respond to the shape properties of objects. So just like in many other experiments, we're going to measure both the serial adaptation from one stimulus to the next, and then look for effects like that. And we can do that and look in these two regions of the brain that we defined by separate means. So within the posterior fusiform sulcus ventrally, we were able to record a recovery from adaptation that was proportional to stimulus similarity. Okay, so again, this sort of evidence of a population code. But within the lateral LOC, this other cortical area that seems to, just as robustly respond to objects and object shape, there really was no or much of any modulation of firing as a function of stimulus similarity. Okay, and we can show this as a bar plot too. This is sort of the recovery from adaptation, the sort of proportional linear effect for the ventral lateral occipital complex and the lateral occipital complex, and there's quite a significant difference between those. Okay, but we can also analyze the data a different way, multivoxel pattern analysis, and here then we're going to take what's the average response to this stimulus across all of its occurrences, and that stimulus, and make another beta map and another beta map, and then once you acquire all of these, you can then create a distributed neural similarity matrix. So this is a traditional MVPA type approach. And so what comes out of this then is a matrix where each cell is how similar was the firing across voxels for one shape as compared to another shape. And this is really sort of the intellectual, it's intellectually quite analogous to what Hans had done in his study of measuring across a set of neurons what was the response to each shape and doing an MDS analysis. So we can obtain these matrices. And so now here again this is the stimulus similarity matrix. So this is perceptually how similar is each shape to the other. And now here's the multivoxel pattern analysis result for, again, the posterior fusiform sulcus, that ventral LOC area, and lateral LOC. And so the result here is just the opposite of what we got for adaptation. okay? This ventral area has a very weak across-voxel pattern that corresponds to the similarity of the stimuli, okay? So there's not much of a distributed pattern in the ventral LOC to match up with stimulus similarity. Conversely, the lateral LOC, which previously had shown, and analyzed the other way, there was really no adaptation effect at all that reflected similarity. Now the across-voxel pattern very well matches the stimulus similarity, right? And again, we can make a little bar plot and show that these two things are significantly different. So the question I want to raise is, what does that tell us about sort of neural coding, or the property of the neurons that are contained within these two brain regions? And so here I think is the take-home message. Voxels in lateral LOC have bulk tuning for particular shape features. And so here's how we demonstrated this. I love these plots, so bear with me for a moment as I walk you through them. Okay, so here's what you do. For a given region, say for ventral LOC, you first go and you take every voxel that's within that region and you identify the tuning function for that voxel. So this is one example voxel, just one, and what's being shown is what was the response in that voxel to every shape. Excuse me. So this is a surface plot, a 2D surface plot, showing what was the amplitude of fMRI response to each, and by the way, this is mean-centered, fMRI response to each of the different shapes, okay? And circled in yellow down in here is the shape that this voxel liked the best, okay? That's the one it responded to the best. So you do this across all the voxels in the region. Every voxel in the region gets its own little 2D shape with a little peak point for the shape it liked the best. And then what you do is you take all of these response surfaces and you align them by their best responding point, right? So you say, okay, for each of the surfaces, line them up so that we place in the center which was the shape that it liked the best. And so then you make a plot of what was the response, or what was the change in amplitude of neural response across the voxels for each step size change of the shape. That might have gone a little too fast. So let me see if I can help you here. So this is a plot which is, if you take this surface manifold for every voxel, and you then align them by the stimulus that they liked the best, and then average them, you get something like this, which is now in the center here, which is not shown because it has an obligate value of being very high, because it's which shape every voxel liked the best. If you then plot like, okay, well, how much did that response decrease as a function of moving away from its most preferred stimulus? In ventral LOC, this manifold is pretty flat. Another way of saying this is that each voxel in ventral LOC liked every shape about the same, right? So every voxel responded about equally to every shape. Contrast that to lateral LOC, which has this curving manifold shape. What this means is that voxels in lateral LOC have a tuning function. There's a shape that they like the best. Maybe they like this shape the best, and they respond a little bit less for the adjacent shapes, and then even less for farther out, and then even less for farther out, okay? So voxels in lateral LOC have a tuning function. They have a preferred shape. Each voxel has a preferred shape, okay? So it turns out like this is a necessary condition to be able to do multivoxel pattern analysis, right? Multivoxel pattern analysis relies upon different voxels having different preferences or selectivities. So it kind of had to be the case that you would find within lateral LOC coarse tuning for object shape, that some things like some shapes better than others, and within ventral LOC, equivalent average amplitude of response to all shapes. Okay? We did some further analyses to kind of show further that what it seemed to be the case is that in lateral LOC there was tuning for kind of coarse object properties, that each voxel seemed to like either oblong things or anyways. We were able to break down a little bit further what are the features of this tuning function, and further able to show why it is that if you have a tuning function like this, you're going to tend to show less neural adaptation. And it's simply just because if you have a change in the shape space, you can move completely out of a voxel's receptive field. And so it just doesn't respond with good neural adaptation to, a stimulus which is shifted completely out of the receptive field of the voxel. But without getting into all those details, because I would like us to have some time for just open discussion, I just wanted to use this figure and the results of this study to illustrate that it's possible that neural adaptation and a cross-voxel MVPA can give different results in different regions. So just because a region doesn't show up with MVPA doesn't mean that the neurons aren't representing something about the objects. In fact, quite the converse, I would argue, that in general, when you can do MVPA decoding, what that is indicating is a fairly coarse coding of the, in this case, object properties. But that when you can do adaptation, now you've found a population of neurons which are able to completely span the space of the stimuli. So that's the final point from the slides I'm going to leave you with. But let's see if we have any questions about that before we go to a general question and answer.
So does it mean that if you find an area with adaptation, you should not find it with MVPA?
It's not an so the question was, if you find an area that shows adaptation, should you not be able to show MVPA with it? It's not an absolute thing. So you can imagine cases in between these where you might have some voxel selectivity or tuning, yet it's still the case that there's enough of a representation of the entire stimulus space within the voxel that it will support the measurement of adaptation. And I think there are examples of that. Russell Epstein, who's a colleague of mine at Penn and has visited you folks not long ago, he just published a nice paper in which he looked at sort of what are the differences in the representation of environmental information as assessed by these two methods, and was able to show there's some regions that show both and some regions that show different, and that even within one region you can find one result for adaptation and a different sort of coding for MVPA. So, and I think there are other papers like this. There's a paper that appeared in Neuroimage just last year that made similar kinds of comparisons. So I think this is something that the field of which the field has growing awareness. But yeah, so I think you can find both or neither or some combination.
And just to follow up on that, it seems like it would depend a lot on your stimuli, right? So if you happen to choose stimuli that lateral LOC was sensitive to, then of course that was, let me say this backwards. If you carefully choose your stimuli and look for adaptation, for example, in LOC, and it's stimuli that
Yeah.
sorry, I'm not flushing this out, but it seems like it would depend on the stimuli. So.
That's right. I think what Eiling is saying is that whether the findings for adaptation in MVPA will interact both with brain region and in the gamut or range of the stimuli that you choose to use and the sensitivity of the region for those stimuli. Is that the flavor? Yeah. Okay? All right, so now we're at a critical branch point. We have exactly half an hour to go, and I am prepared to talk about de Bruijn cycles. So, but I know there are a number of folks in this room who either do functional MRI or are planning on doing functional MRI or have at least read a functional MRI paper, and I'd love to spend this last half an hour just having a bit of a discussion and an exchange about different topics in functional MRI. So does anybody have any questions that you'd like to ask or discuss? And I will say that there was an entire break period to think of things too. I remember that when we first started out, there was somebody who wanted to ask about motion correction or the effects of motion. I don't know, maybe that person finally gave up and left, or maybe that was you had asked that? Okay, all right. So we could, is that, okay, motion? Yeah, let's try and make a list, and then we can, so let's do a little brainstorming first. Okay, motion correction. Any other topics in fMRI?
Finite impulse response functions.
Okay. So you're interested in like FIR modeling of fMRI data and then using that for looking at hemodynamic, as a substitute for hemodynamic response functions. Yeah, and I think we could even broaden that out. Then that moves us into the realm of talking about basis sets. So basis sets of covariates to model data. Okay, so that falls in sort of the stimulant data analysis side of things. Any other topics? Yeah?
Single trial based on FIR models to measure dynamic connectivity. No dynamic connectivity and dynamics within the area. I mean the classical work by Ravi Menon.
Yeah, right.
That they saw that the motor cortex is driven by the visual.
Oh, trying to look at timing between regions.
Yeah, and then looking at so you estimate the parameters using a FIR model, and then going into the time to peak or the amplitude, and there is also another work by the Dennis group.
Yeah. This
recommended by Bradley Buckner.
Yeah.
To measure based on the FIR model parameters. How can you infer
timing? Yeah, I think you're asking, this sometimes gets described as stimulus onset asynchrony designs. That's like one term that gets applied in this. I think I know that. Okay, so that also falls in like the in the analysis and modeling of data. And any final topics?
Expectancy versus rotation.
Oh, okay. Well, then I might hand the pen over to you for that. I mean, if you have some thoughts about it, I don't have prepared remarks on the topic, but I'd be interested in your thoughts about expectancy. Oh, I see. So, like what might be relevant to some of the things we were discussing before, what might be the relationship between what appears to be adaptation as well as in contrast to a predictive coding account of adaptation. I think it's, that would be a great thing for us, I think, to talk about over a cup of coffee. Because we're going to take pity on the audience for adaptation because we've talked enough about that. Okay? Is that it? All right. Well, look, I'll tell you what. So motion correction, that falls within the realm of just data preprocessing in general. So fMRI data undergo typically a number of preprocessing steps prior to the application of statistical models. And so some of the things that are included in there is sync interpolation for slice timing. There's motion correction, spatial normalization, smoothing, and so on. So let's actually just apply just have a basic understanding of what these things are, and then we can talk about what some of the issues are that come up in it and what order in which to do them, because this is often a topic of some debate too. Okay. Let's start with motion. So, it's a shame. People move, right? So you put people in the scanner, you pad their heads with foam. In the old days, at some places, we used to have bite bars where the subject had to, so they had to bite onto a plastic thing which would fit to their teeth, it held their head still, but the experiments had to be brief because people hated it. But despite everything you do, people move. So the question then is, what will be the effects of motion upon your data? So in one sense, in principle, this could be a really easy problem to fix, right? So if it were the case that the brain was a rigid object and that you got an instantaneous picture of the brain at different points in time, then correction for motion would be just a simple matter of aligning the data back, aligning the brain back to its original point in space, right? So here's the brain at time 0, and time 1, and time 2, and time 3. And it turns out that for a rigid object, there are 6 degrees of freedom, or 6 parameters, that can be calculated to align it back to its original state. Okay? And those 6 parameters are, left and right, up and down, forward and back, and then pitch, roll, and yaw. Okay? So these are the rotations and translations. So in principle, that should be pretty easy. You have an image of the brain at each point in time, and you just calculate by a least squares minimization how you then sort of line that brain back up with its original point. And so that's what's done as a standard part of data processing in fMRI, is there's a calculation of those 6 parameters and then a resampling of each brain image back to its original point. Unfortunately, that does not completely fix the problem. And it does not completely fix the problem for a couple of reasons. So one is that we do not obtain instantaneous pictures of the brain. Okay, so it's not the case like we don't have like a camera that we take a flash and poof, there's the picture, right? Instead, we obtain the picture with a camera as if it had a long exposure. So it's like picture, 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 like that. So if there's movement during the time that the picture's happening, just like with a photo you take, you get kind of a blurred picture. Moreover, the way that that picture is obtained in fMRI is by slice. So we get one slice, one slice, one slice, one slice, one slice. Okay? So movement, so if you had the brain in one position halfway through taking the picture and then it moved, then the slices are going to be all intermixed and the data will be sorted in a way that a simple linear correction does not fix. So that's one reason why motion isn't completely fixed by realignment. The other component is something called spin history effects. And here's the idea. You know, the way fMRI works, remember, you're in the magnetic field, it's radio frequency energy coming in, and then signal coming back out, right? And so what we're doing is, when you do fMRI and you have the images coming one after the other, the protons are going like, oh, 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 right? Because you keep getting the energy comes in, then they relax, energy comes in, then they relax. And the speed with which those protons relax and the sort of radio frequency energy that they absorb depends upon where they are in the magnetic field, which we recall varies. So if you have a proton which just goes, oh, and then moves to a different place, it's now relaxing in a different magnetic environment than it was originally. And these are what are referred to as spin history effects. They create signal changes in the data that is a completely nonlinear interaction of the movement of the head and the magnetic field and all the rest. So because of these reasons, because simple realignment doesn't completely fix the motion, it's common to actually include a covariate that models motion and then include that with the data as well to try and account for variation in the data that's explained by it. Anyway, that's motion. Sync interpolation is the correction that's done because the image is acquired as different slices. We try and project back in time when each slice was acquired to kind of get them to line up as if the whole image was acquired at one point in time. So that's a standard step as well. Spatial normalization is just taking a brain and then aligning it to some standard anatomical reference frame. And there are both volumetric and surface-based approaches for that. And finally, spatial smoothing. So spatial smoothing, it's an interesting topic. So, you know, we go to great efforts to acquire fMRI data with really tiny voxels, and then we'll often go and then smooth the data in space. This is done less nowadays in MVPA analyses, but it's still quite common when you're looking at direct effects or adaptation type things. So why does smoothing make sense? Well, smoothing can be valuable for, in a sense, two reasons. One is that, you know, the brain is a big place made up of lots and lots and lots of little voxels. And as we've talked about previously, if you're going to do a statistical test across the entire brain, you have to account for each of the tests that you've done in each of these locations. And so that then requires a p less than 0.000001 or something to correct for all of these components. But if it's the case that by smoothing, you kind of make the brain have less independence, each voxel now has kind of less independent component, then you now have fewer independent measures you need to correct for, and you can drop a few of these zeros off. Okay, so that's one purpose of smoothing. Another purpose is aesthetic, but I think really the main reason is that you can, as a rule of thumb, the smoothing that you pick should match the size of the thing you're trying to detect. So if you're trying to detect something that's, 2 centimeters wide, you should smooth with a kernel that's 2 centimeters wide, as a general rule of thumb. Okay. Were there any specific things in motion then that you wanted to talk about, or does that kind of cover it?
I would like to just ask you whether among the many hardware techniques which are used to predict and to correct motion, what would you reckon? We have optical methods, we have methods which involve microcoils and tracking of the positions of fiducial markers. And there are other even accelerometers which can be used. But of course this is I mean, this simply is for the moment very experimental. Do you think it's something which is going to be emerging solution?
Yeah. So the question regards, of the different techniques, there are further techniques that can be used for trying to measure motion and correct for it. It turns out that Siemens scanners implement something called prospective motion correction as well, so the pace correction. You can place, as you mentioned, fiducial markers, basically little beads on the surface of the head, and then measure the displacement of the beads. So I think in general, for studies that are being done on like a normal population, so if you're studying cooperative 20-year-old people, extra effort to correct for motion is not worthwhile, because generally you'll either have subjects who hold still and you'll keep their data, or if you have a subject who moves a lot, you throw away their data and just get another subject. For studies which involve kids and adults and older folks or rare populations, right? So people who there's a limited number of people you can study, patients or otherwise, then these extra efforts can be worthwhile. I've found fiducial markers are useful for studies in particular where you're expecting big brain activations. So if you're doing like a language task or like presenting stimuli that flash the whole screen, because large neural activation can be mistaken for motion by some of these, by this motion correction calculation. So by having a fiducial marker, you can get a measure of motion that's not contaminated by neural activity, right, by task activity. So of those, fiducial markers are the only ones I've tried. I don't know about the effectiveness personally for the other ones. But I think fiducial markers have the nice feature that they're easy to apply. They don't require any longer time scanning. And then you can get a true measure of motion. Okay? Yeah.
I wanted to ask you about your opinion, because there are methods, analysis methods, that don't need smoothing. I mean, if you apply the Bayesian approaches that they consider a spatial model for your data, you don't have to do smoothing. But they are not applied in the literature. I mean, they are available in SPM and the software packages, but people don't employ them. Do you have a risk? I mean, do you have a thought about that?
Oh, about why these
These advanced techniques that could solve these problems.
Right.
Smoothing, for instance, or for like in FSL, in AFNI, and in SPM, they have different ways of dealing with the serial correlation.
Right.
And these advanced techniques, apart from dealing with the spatial correlation, they also deal with more advanced with more advanced models of the serial correlations too.
I see.
So it's
Okay. Okay.
So
I see. The question is, there are, okay, so I think your question regards both smoothing and then also how one interprets or uses the non-independence of data across space and then calculation of statistical thresholds and things like that. Is that what you're referring to primarily?
yeah, but also for fitting the model. I mean, when you fit the model, you can assume spatial model.
Okay, got it. Okay, so
all these posterior probability.
yeah, okay, I see what you're asking me. Okay, so the question that Cesar is referring to, or the question that it brings up is, fMRI data has traditionally been analyzed in this what's called massively univariate manner, which is that for each little point in the brain, we're going to now fit a general linear model, right? And so we'll calculate a statistic for every single tiny point in the brain. And so there's been a whole set of methods that have come along from using that as an approach. But from the earliest days, it's been obvious that there's spatial coherence present within the data. Even beyond what might be described as smoothness, spatial coherence is the observation that, hey, the signal here is correlated with the signal here, right? So that's nothing that can be described just by simple local smoothness. This is actually sort of long-range correlations in the noise, what we now think is very interesting about resting state data. So there are techniques that you can use that explicitly take advantage of that. But as Cesar mentions, like they're not greatly used or people haven't used them very much. And I don't know, why is that? It's not because they're bad or not good. I think it's just that most experiments that people do nowadays are ones, again, where it's across subject studies, where all of these problems drop. A lot of these problems drop away when you come to the second level. Like all you're doing is you're estimating an effect size in each subject and then bringing that map up to a second level analysis across a population, in which case a lot of this stuff doesn't really matter so much. And then I think just as those methods were reaching some degree of maturity, MVPA came along. And so now that's like the technique that everybody wants to use that is multivariate in its approach, right? Looking across the cortex as opposed to a single region. So I think it missed its shot because a little bit too late for single subject studies, of which there are still important single subject studies going, but and too early for MVPA.
So we have to blame MVPA.
Yeah, that's right.
It's great.
MVPA. Okay. So, and then now if it's okay, let's talk a little bit then about analysis of data and experimental design. Okay. So we did not spend a lot of time in the first 2 days talking about this, but the general principle of functional MRI, or the way the data are often analyzed, is assuming this linear model of the hemodynamic transform of neural activity into bold fMRI signal, right? And so that's the basic idea, that you can have a pattern of neural activity like this, and if you know something about the linear system that is bold fMRI, by a process of convolution, you can predict that the response should be like this, right? And then that now is a covariate that is then fit to the data in each voxel of the brain, right? So you just try and say, how well does this model fit the data? So that's fine, but there are a whole realm of experiments and situations where you might not be confident about some aspect of this neural model, okay? So this whole approach begins with the assumption that you know exactly when these neural events took place and what the shape of the neural events were, and that you know what this transform is like, okay? And that might not be the case. So there are many ways of adding additional covariates or parameterizing the space of possible responses to fit the data more flexibly. And this is the realm of what are called basis sets. So a basis set is a set of covariates that have the property that as a collection or in combination, they can model variation in possible response. So they can fit to a space of possible responses. So one kind of limited or constrained basis set that's used is oftentimes the hemodynamic response and its first derivative. Because, and now I got to draw this, and I suck at drawing this. So something like this. There, we'll call that. So let's suppose this is the first derivative of this, okay? A property, and this has to do with the fact that the hemodynamic response function is a continuously differentiable function. And anyway, by something called the Taylor expansion, you can, this covariate, which is the first derivative of this one, is able to model shifts in time of the neural response, okay? So suppose you weren't exactly sure when the neural response was going to have its onset, either because it could occur at different slices in the brain, which is being imaged at different time points, or you just don't know exactly what the delay is going to be between presenting your stimulus and some neural event in the frontal lobe, or because the shape of the hemodynamic response for your subject looks more like this as opposed to like this, okay? So any of these differences that might result in a difference in timing can be modeled by or accounted for by this first derivative, okay? It's going to account for any differences in timing between this modeled response and the actual response. So that's one kind of very simple basis set that turns up quite frequently. There's something else, which is, let's suppose you wanted to discover what the shape of the response is over time. You didn't want to assume that it was a particular hemodynamic response function. Well, the way you can do that is, so you have some data, which might, look like this. Okay? And you don't want to have to assume what that shape is. You could model it with a set of covariates that look like this. So one that is like this. This is time point 0 after the stimulus. And then one that's like this, which is time point 1 after the stimulus. And time point 2. And so on. Okay, so this is time equals 0, 1, 2, and so on, up to say maybe 16 seconds. And so what the properties of these covariates are is that each will be scaled up or down to best fit, this is now the data up here, to best fit this data, right? So you would then have a fit to the data. The model would fit the data, and it would look, like this. Okay? And so then the loading on each of these covariates you can take to then create what was the average response across all the times the stimulus appeared. Okay? And this approach is referred to as an FIR model, or a finite impulse response model. Okay? It also has a lot of other names. It's a shifted set of Dirac functions, which is another term for an impulse, or a shifted set of delta functions. But this is just a set of covariates that can flexibly model any response that is, it will flexibly model the average or central tendency of a response across trials. So is that the kind of thing you want to get at, or do you have a more specific question about FIR models?
Now the next step is, can you assume that the activity you have on T1 or T0
Yeah.
is independent of the activity of T1?
So, okay. So then what's the relationship between these two things? Can you assume that the activity at T0 is independent of T1? Okay.
Can we pick up something in this first window that actually belongs to the second window?
For the same stimulus onset for all the trials, or with asynchrony? Because it's different.
Yeah, right. So that question, now the question is like, okay, so can you then somehow distinguish what was the, and now we're talking about the hemodynamic response function, we're not talking about the neural function. So maybe what you want to do is you want to model, there's a stimulus, and you'd like to know what was the neural activity at time point 0, time point 1, the actual neural activity, right? That's actually a fundamentally different and difficult question to ask. So in practice, because of the shape of the hemodynamic response function and its filtering properties, that you cannot distinguish what was the response at the neural activity at 1 second after the stimulus versus 2 seconds after the stimulus, right? You can flexibly model what was the shape of the evoked bold fMRI signal, but you have no way of going back to recover this. Although I was introduced just yesterday to some interesting work that Cesar is doing in trying to model or predict what would be a likely, in a probabilistic sense, set of neural spiking events that could be responsible. So actually, the guy who is best equipped to answer your question about trying to model subtle differences in the amount of neural activity in time is right next to you. So that's really something that he's spent a lot of time thinking about. I will say as a general principle, though, this involves inverting a model where there is insufficient information to do the inversion, okay? Or deconvolving where you have had information lost. So to a first approximation, the actual pattern of neural response, or the timing of neural response in the order of seconds, is lost forever. You can't get that back. The best you can do is separate out neural events on the order of about every 4 seconds. Okay? And that can be done with a set of shifted hemodynamic response functions modeling an event. And so this is something that was quite popular, and I think is still used in a number of working memory type paradigms. And so this is the idea that there is a stimulus on the screen, and then there's a delay, and then there's a response. And suppose that this delay is like 8 seconds long. Okay? So it's like remember, this color blue. And then you got to remember and remember and remember and remember. And now you need to make a response. And what you would like to do is determine, is there any neural activity in the delay period which is separate from the stimulus period? Okay? And that can be done by creating a neural model which is like, okay, this was the stimulus, and then this was the delay, and then this was the response, right? These are 3 separate neural models. And then convolving these with HRFs, with response functions, and then seeing how well they model the data. It turns out actually, technically, you don't use a square wave for this. You use something which is peaked here, but we don't need to get into that detail right now. But in practice, your ability to discriminate closely spaced events like that decreases very quickly as you get shorter and shorter time scales. And 4 seconds practically is about the limit of where you can do that. Okay? Okay. So we have 3 minutes left to describe stimulus onset asynchrony designs. So I think I can do it. So the idea of these kinds of experiments is what you want to do is measure the difference in timing of neural activity. So now there's 2 different things actually. There's the difference in timing of 2 different neural events, and then there's the difference in timing across brain regions. And these are 2 very different kinds of inferences. So here's one sort of experiment. Suppose I present a stimulus, like it's a tilted letter E, and you need to make a response. Is that a letter E or not? Okay, and the letter can be either tilted, and so then you make a response, or the letter can be upright. And you have this belief that there is a delay, a mental rotation delay that takes place before you recognize that it's the letter E, so that within an area of the brain that represents letters, down here, this doesn't get the identity of the E until later in time, compared to here. Okay? So there's a slight delay in the onset of activity at this location for the tilted E relative to the upright E. Okay? So what that might look like is if you took all the tilted E's and all the upright E conditions and then took the data from here, you might find over time, here's when the E was upright, that's the fMRI response, and then here's where the E was tilted. Right? And you'd be interested in claiming that difference in time to peak represents a delay of neural activity getting down to this location because of some mental rotation process. Okay? Turns out that's a perfectly valid experiment, and that can be done, and people have done this. It turns out your power to detect about 100 millisecond difference in timing is about equivalent to, it's like 10% of the power that you have of detecting like a button press in motor cortex. So it's not terrible power, actually. So you have about, for 100 milliseconds, you can detect that. You'd need about 10 times more data to detect 100 milliseconds difference than you would need to detect a button press in motor cortex. Okay? So definitely within the realm of measurable. And people have done this in the visual system. You can present flashes of light and delay them and all the rest. So the reason you can do this is that the hemodynamic response function for this region is always the same, or at least you're able to assume it's always the same. It isn't changed by the underlying neural activity. So you can work from this back to these 2 possible neural models and compare them. Here's what you can't do, or at least here's what's much more difficult to do. And that is say okay, I presented 2 sorts of stimuli. I presented just this letter E. And I'm interested in showing that there is a neural response here in the parietal cortex that happened before this. Okay? And, or actually we'll say down here before up here. And this is the data you get. So in red, that's from the area down here, and in blue, that's from the area up there. And you want to claim, look, the neural activity came on sooner down here than up here. Okay, that's the claim. You want to claim, look, this means earlier neural activity here than here. The reason that might not be a safe conclusion is what if the shape of the blood flow response was different between the 2 areas? And there is some evidence that can be the case. So that is just like a broad introduction to that topic. I'm sure Cesar has a more specific instantiation that he wants to ask me about. But I will say, we are now right at noon. You guys have been an incredibly patient audience over more hours than I care to add up. So I really appreciate you guys coming out and listening to all this, and it's been my pleasure to be able to come out here and join you. And I hope I find a chance to come on back and visit Eiling sometime. So.
Let's thank Jeff again.